mediated immunity is difficult to measure. Making antibodies that are antibody tests that are specific takes time, and there was always the concern about cross-reactivity with other coronaviruses. And that ultimately, the calculation of what we call the R0, which is if you have someone who's infected, for every person infected, how many unexposed or unimmune people will be infected subsequently. And the initial estimates was the R0 was in the 2 to 3 range, 2 to 4 range. And in order to actually do that calculation properly, you do have to know what the numerator and the denominator is. So what has worked in British Columbia? So we have a government that used evidence-based approaches, and we can be thankful for that. I think we see what's happened when the governments totally ignore this. We did have Bonnie Henry, who I think was a fabulous leader for how those messaging were released to us. The BC CDC exists in British Columbia, and it's really an integrated public health facility with close regional ties, including the First Nations Health Authority. And you had a public health laboratory that quickly implemented and deployed nucleic acid tests to frontline labs, including life labs. And that actually gave us a lead time of about 4 to 6 weeks based on comparisons with other jurisdictions and the other jurisdictions which required a commercial vendor. One thing that we didn't have was this ability to integrate data in real time. And this is the dashboard as of the 3rd. This dashboard pulls data in real time on the cases, the number hospitalized, the number in the ICU, the confirmed deaths, the recoveries, and also the turnaround time for the tests, and a whole bunch of other features, which really is pretty impressive. And I can tell you that the infrastructure to bring this together is still cobbled together, but had you wanted to do this without this COVID epidemic, pandemic, it would have taken another 5 years. So this was rapidly pulled together. And then there's other versions of this which actually understand the capacity in the entire province for every jurisdiction that's doing testing. So in terms of the nucleic acid test, so when you produce a test, the sensitivity and the specificity of that test is really based on how that test performs. But the predictive value really depends on what's going on in the population. So on this figure from the New England Journal, and I hate this figure because it's very confusing to read, but it is from the New England Journal. If you look at the x-axis, the chance of infection before a test result is known, what's your pre-test probability? And essentially, if you use a test that's less sensitive, you're going to miss ones. And if you use a test that is poorly specific, you're going to get false positives. And so this relates to different scenarios for a test that is 90% sensitive with a specificity of 95%. This is a test that's 70%, and there's this literature about a 70% sensitivity. And this gives you that chance of infection given a negative test. So the point of the matter is if you use an insensitive test, then you can get a false negative. And again, if you use a poorly specific test, you get a false positive. So I hate this figure, but I just want to summarize here. We know that there's this literature about pre-symptomatic infections. We know that there's this literature about asymptomatic infections, which is the bane of my life, and I will show you that shortly. But ultimately, most of the onward transmission, I do believe, if you tease apart all of this literature, which is pretty difficult to interpret, most of it is due to symptomatic infections. The nucleic acid tests that are done in Canada and British Columbia are probably in the 90 to 95% sensitive. It's clear that in some cases, the respiratory sample from the nasopharynx will be negative, and sometimes they'll have a severe lower respiratory tract infection. The specificity is probably in the 99.5% range, and that's an estimate, but that means that if you're testing in a very low prevalence situation like we are in now, for every 200 people, you're going to get a false positive, and getting a false positive isn't a trivial challenge because it creates a whole bunch of angst. So there is still a small chance to miss an infection early on, and with low prevalence testing, you would have a greater risk of false positives. So this is a very interesting paper that came from many in Nature Medicine, recently published, and really looks at a very interesting scenario between what is the receiver operating curve and what is the area under the curve. And it looks at people from both the UK and the US, and these are ones who tested RT-PCR positive, so they're NAT positive. And what it illustrates for you is that for those people with symptoms, the correlation with symptoms and positivity is in the 75% range. And that illustrates to me that most people who are going to be infected and infectious are likely to have symptoms. And here's the controversy in the literature. So there was a recent publication published by Oren from the Annals of Internal Medicine, looking at a whole bunch of these outbreaks where you have a bunch of people who were positive but asymptomatic. And what's clear is when we look at our healthcare workers in the long-term care facility, very few of them were asymptomatic. Some of them were afraid that they had to come into work. Similarly, with the elderly in a long-term care facility, that's clear that people who are elderly in a long-term care facility, many of them can be asymptomatic. If you looked at the poultry outbreak, many of the workers who claimed they were asymptomatic, when you actually posed the question to them afterwards, they said they just were afraid not to come into work because they were the breadwinner. So I will say that asymptomatic infections do occur, pre-symptomatic infections do occur, but how important they are for onward transmission is challenging to articulate and to deal with. Where are we for serology? So this is a publication by Kelman Barkley, and essentially provides some insights that it usually takes about 10 to 14 days after clinical onset of disease to develop antibody. And with this virus, it's very strange that you seem to make an antibody to M and G and A almost all at the same time. And what is clear from this figure is people with more severe disease are more likely to get a higher antibody level, and people with asymptomatic or limited disease tend to be a lower antibody level. So this is a challenge. We did a lot of work trying to understand point-of-care tests. And essentially, we found them unreliable, largely because we would get some kits that looked one way on day one, a separate lot looked like a totally different kit. And so I think while point-of-care kits can be used and work, they need to be quality assured to a degree that we haven't seen yet. We've done very extensive comparisons collaboratively with St. Paul's and Children's and Women's, and the commercial major vendors produce very good assays. Their specificity is high, greater than 99.5%, and their sensitivity, probably at 21 days or beyond, is probably in the 95% range. So when you look at the uses of these assays, well, clearly one of them is to look at the seroprevalence, and we've just done some seroprevalence work, and based on the preliminary data, we have a seroprevalence in BC of about 1%. And we'll talk about that because that means there's a lot more people who can get infected. One of the biggest questions is are you seroprotected? From non-COVID coronaviruses, the evidence is you do have some degree of protection, but that durability is unclear for many of them. If you are a rhesus macaque, there's some data that you will be seroprotected. And there is no data at this point that of people getting reinfected when they've been infected already. There was some bad literature about potential reinfections. So at this point in time, this needs to be studied, and we're trying to do that collaboratively across the country. And then in terms of its clinical utility, we know that it's associated with some inflammatory syndromes in children, these things called COVID toes, which are probably an immunological response to the virus. And then there was some push to use this for clinical diagnosis, but there was a recent Cochrane review that showed that about at 7 days after infection, the sensitivity of serology is in the 30 to 50% range. By 8 to 14 days, it goes up to the mid-70s. And then you really do need at least 3 weeks. And by 3 weeks, you're either better or you're no longer infectious. On this figure also shows you another headache that we have in the serology realm. So this is a small study from Nature Medicine, but it looked at what happens to the IgG serology, positive in blue, negative in, I guess that's green. And essentially during the acute phase, you have very similar percentage reactivities. If you follow them out in the convalescent phase, what you find is the people with symptomatic infection, more severe infections, they tend to have higher antibody responses. And those who have asymptomatic disease, some of whom could be false positives, tend to be seronegative or lower seroreactivity. In some studies from New York, up to 5 to 7% of people who were infected never mounted an antibody response. So I realized that people are keen to have their antibody measured despite the lack of evidence for substantial and durable immunity. And I think the most important message is until you can show that seroprotection is documented, these tests should not be used to guide infection control decision-making, the use of personal protective equipment, the timing of return to work, and physical distancing policies. Herd immunity. So herd immunity is that term that says, okay, well, if you've infected enough people and they're immune, and I've just shown you that that isn't a simple question. If you have enough people who are infected or enough people who've been immunized and have immunity, you can stop transmission. So there was a recent publication in Science where they assumed this R naught of 2.5 and looked at age in community and how communities interact with each other. And they suggested that for this virus, the herd immunity level could be at 43%. And I just told you that we're likely at 1% in British Columbia now. And then there was a recent publication from Spain that even where they had massive infections, the percentage infected is in the 5–6% in general. So classic herd immunity typically is in the 60%. So here you have the challenges of creating these models. And of course, no one knows whether it's 43% or 60%. And so we're going to have to look at this and watch this over time. I just want to mention a little thing about genomics. We were involved with using real-time genomics, and Richard Harrigan and Natalie Prestojecki have done an excellent job at basically doing whole genome sequencing. And they've sequenced about 1,000 genomes that have circulated. And essentially in British Columbia, the B1, which is this dark green, is the most common viral strain, if we'll call it, that's been circulating. And it probably links to sequences that have been derived from European strains. And then the second most common are this A1, which is this sort of teal or red group. And those correlate better with sequences that have come from the US. And then there's a bunch of other sequences that were originally derived from China, but really haven't continued to infect British Columbians. So where genomics can help us is it can tell you if you have ongoing community transmissions. or you have repeated introductions of new strains from travelers, whether you have the multifocal transmission clusters that are occurring. And just remember that transmission across health authorities, this virus doesn't care about our provincial borders, nor does it care about the international borders. In terms of treatment, there's a lot and very little at the same time. There's these ACE receptor efforts, nothing's been clear. Hydroxychloroquine comes in and out of the news, but is probably not going to work. There's efforts with this IL-2 receptor activation prevention monoclonal, and essentially this may have some impact to reduce the cytokine storm. There's lopinavir, which was used for HIV, which really has limited benefit. And then remdesivir, which has been basically purchased by the US and pulled the whole market, reduces the replication and may have an impact, but I'll just show you, if you look at this publication from Bagel in New England Journal, you see those curves aren't really spread apart between people on remdesivir and placebo, and clearly the benefits are likely to be marginal. What people have learned is avoiding intubation is a good idea if you can do that. And dexamethasone, even though it's been reported to be important in reducing mortality, I actually wasn't able to see any of the raw data to support that. And I think we have to see that raw data and figure out what to do, because if you use dexamethasone, you're going to immunosuppress people. And so there's a lot more to be learned there. In terms of vaccines, again, an incredible effort to produce vaccines, including some being developed in British Columbia. But there's these RNA vaccines that have not been used generally, DNA vaccines where there's been some studies, there's recombinant protein vaccines, there are these vectored vaccines, some of which are adeno5 vectored, and that means that people who had adenovirus previously may not mount a good immune response. Inactivated vaccines and live attenuated vaccines all being worked on. The time frame, this is going to be tough. Remember that if you're going to use the vaccine, you're going to need to make sure it's safe, because most of the younger people aren't as severely affected by this virus. The older people typically don't mount a good immune response. And so you're going to have to be really careful about the safety. And so there's lots to learn there. This is some modeling that takes place at the BC CDC and has been reported by Bonnie Henry. And it really does talk about this notion of this R0. And keeping this R0 under 1 is really the way that you can prevent massive spread. And essentially, this is where we are now in British Columbia. And if you have these large conventions and things like that, with a lot of people congregating together, you can easily push that up. And this is going to be our challenge in the future. How do you maintain this and at the same time deal with the other issues from society? And this also is a model that really looks at, again, this notion of trying to keep your R0 low, and that looks at the physical distancing and the time to contact tracing. So in British Columbia, typically people are contact traced within 24 hours. And again, as you have weaker physical distancing, you actually need to be even more aggressive with this kind of work. In another area which we all recognize is the untold effects of this pandemic, articulated by Rosenbaum in New England Journal, articulated in the Canadian Medical Association Journal. The impact on kids and children is huge. The province and the BC CDC have been looking at this in terms of classifying people in terms of what are the essentially the the unintended consequences and really trying to think through them both from a health perspective and other sectoral harm, with the need for immediate action and response, 6 months, 6 to 12 months, and whether you monitor them. And we all realize a lot of this is access to health services, food security, housing, shelter, education, income, employment, economic growth, and you've heard about how much in debt our country is going to be. So here's my crystal ball. And I do believe infection will lead to some protective immunity based on how other coronaviruses perform. And I think some of that will be measurable by serology or antibody-based systems. There was a recent paper out of the Karolinska Institute that looks at cell-mediated immunity, which may be another mechanism of really understanding the immunity that you develop. Anyways, this is going to take some time to study. If achieving herd immunity requires 40 to 60% to be infected, even in highly affected areas like Montreal or Toronto, there are many more to infect. Certainly in New York, where people have suggested that 28% of the population has been infected, even more can be infected there. Fast-tracked vaccine needs to be safe. And remember that vaccines don't often work well in the elderly. Maybe if you vaccinate all the young, and then the elderly will not be exposed, that's a good strategy. But remember the complexity of doing this on the fly. And then finally, I'll leave you with this comment that living in a bubble and avoiding infections has consequences. Our body and our immune system works on a use it or lose it principle. So the notion that you're going to protect everybody by us being in a physical bubble for the rest of our lives isn't going to work because our immune system needs to be primed for this and other agents. So given COVID, I call it the pandemic's pandemic. And I will also say, take everything I say with a grain of salt because the literature changes by the second. And then I just want to end with this comment that BC's life sciences really provides us some fabulous intersectoral opportunity. Simon Pimstone has been working closely with Genome BC and others to actually try and figure out how you can safely bring people back to the workforce, engage the staff in the workforce, and create a monitoring tool that you can actually make some evidence-based public policy. So why don't I leave it at that, and I'm open for a few questions.
Thank you very much. That was, as always, incredibly informative, and you have given us a little bit of a history and different perspective on how we've lived the last 3 or 4 months, and you know, taken a little bit of the fiction and replaced it with solid facts, which we really appreciate. Your leadership and those of the BC Centre of Disease Control and Dr. Bonnie Henry has been extraordinary, you know, through this pandemic, and we all are extremely grateful to live in BC and have people like yourself at the helm. Obviously, it's a little bit of a confusing time in that we've had some questions which I'll probably forward to you after, just in the spirit of time. One question I think is on everybody's mind, and you really addressed it in your last slide, which is that you know, we've been chasing science for 4 months. We shut down our economy to try and ensure we had the capacity within our healthcare system and to understand the science. And as you have told me personally and very adequately demonstrated today, there's still a lot of science to understand. And as you look ahead over the next 6 months, everyone's preparing for the second wave. How can the average individual prepare for the second wave, and should people be setting their expectations that they're going to continue to be working from home, yet trying to find that balance, as you say, of living in a bubble isn't necessarily the best way to protect our immunity on other issues. So what do you see, and what advice do you give to people as they think ahead over the next 6 months?
Yeah, and thank you, Wendy. And so I think about it in the following way. Often what happens in Australia portends what happens in our part of the world. And in Australia, you see that they were containing it, and now they've recently shut their provincial borders. And I think that you can also see that if you don't pay attention to this, you also have massive challenges. So even in the US, where half of the population says, ignore this, and the other half of the population says, I'm scared. Like, it's almost as if you totally ignore it, you're not going to solve the problem, and you're going to hurt your economy as well. And so I think really the way to think about it is, it is possible with controlling the R naught through some physical distancing and things like that, that you can bring your economy together. And if you do that carefully, then I think you can avoid a very powerful second wave, because what we do have in place in British Columbia now is this ability to have real-time data of what's happening. And that really it's about us working together to think through how you can prevent these massive infections. And essentially what we would be able to do is try to respond quickly to prevent large-scale outbreaks. Because once you have a large-scale outbreak, the only way to handle that is by these lockdown kind of procedures. So it's not a trivial issue, not simple to do, but everyone's going to try and figure out how to do it together.
Yeah, absolutely. I think one of the really interesting things that has come out of this, and a benefit is, and you talked about the importance of data and the push of being able to get the data together quickly, which I would assume is going to put us in a much better state moving forward. And as many people know, I have a data background, and I've actually been quite excited to see how data has been able to be more accessible, consolidated better, and there's a whole conversation and a different webinar that we'll have about contact tracing at some point. But it is wonderful to be able to have you have and many that are leading this have the access to data to be able to really understand what's going on in a timely basis.
Thank you.
So thank you very much. I know you need to run, and we all, on behalf of everybody, we all appreciate everything you are doing and what everybody's doing at the BC Centre of Disease Control. So with that, I'm going to turn it over to Joe to introduce our next speaker. Thank you.
Thanks, Mel. We really appreciate it. Take care. So hi, everyone. It's now my pleasure to introduce Dr. Brad Nelson. Brad is a native of Vancouver and received his BSc from UBC in 1987 and his PhD from the University of California at Berkeley in 1991. He worked for 12 years at the Fred Hutchinson Cancer Research Center and University of Washington in Seattle. He is the founding director of the BC Cancer's Dealey Research Center in Victoria and a professor of medical genetics at UBC. He is co-leading a clinical trials program focused on the use of genetically engineered T cells for the treatment of leukemia and lymphoma. In 2019, he co-founded Innovakine Therapeutics with Dr. Martin Boulanger in collaboration with Zymeworks. Innovakine has developed a platform technology that will allow clinicians to send exclusive customized signals to immune cells anywhere in the body with broad clinical applications for cancer, infectious disease, autoimmunity, transplantation, and regenerative medicine. Thanks, Brad, for joining us today. We appreciate you making the time.
Well, thanks, Joe and Wendy and Ryan for inviting me to present on behalf of Marty Boulanger, who's on the line, but he's got a somewhat unreliable Wi-Fi where he's at. So I'll do today's presentation. And there goes my phone. I'm going to be talking about Innovakine Therapeutics, which, as Joe just indicated, what we've developed here is a platform technology that will enable safer and more effective cell-based therapies. So I'll just start with a picture of myself and Marty. Again, we're the two co-founders. We launched Inovokine last August, so coming up to our first anniversary in about a month. My day job is to work at the BC Cancer Agency, where I'm co-director of the immunotherapy program. And Marty works at UVic. He's a Canada Research Chair and a professor of biochemistry. And Inovokine really brings together our two areas of expertise. I'm the cancer immunotherapy guy, and he's a structural biologist and protein engineer. And as I explain our technology, you'll see how those two things come together to form Inovokine. So we're talking about cell-based therapies, just to get everybody warmed up to that idea. What this means is instead of the traditional concept of treating people with drugs, treating them with living cells. And so, of course, transplantation has been with us for several decades now. What we're talking about in 2020 is the idea of taking that a step further and using genetically engineered cells. And as we'll discuss, particularly in the area of cancer, with CAR-T cells, this is revolutionizing treatment strategies. And we and really the global community see great potential, not only with cancer, but with applying this sort of next generation of cell-based therapies to autoimmunity, transplantation, and regenerative medicine. But a key challenge that comes up when you treat a patient with cells is controlling the behavior of those cells once they're back in the patient's body. Again, these are living entities, and with their own properties, their own behaviors, and we'd like to gain some degree of control over that. So to fully realize the promise of cell-based therapies, we need a new generation of molecular tools that will allow this precise control of cellular behavior. And that's where Inovokine comes in. So we'll use the example of CAR-T cells, and you'll see how our technology can apply to this. And this is really the breakthrough that's happened in the last few years in the cancer immunotherapy field that has really ignited interest in cell-based therapies. So, and in particular, what's working is CAR-T cells directed against an antigen on leukemias and lymphomas called CD19. So the way CARs work, here we've got a tumor cell with an antigen, CD19, on its surface. And what a CAR is, it's a chimeric antigen receptor that's a half antibody on the outside of the cell, which can bind to the target. And then the other half is derived from the T cell receptor complex, and so generates activation signals for the T cell. So the net effect then is that when the antibody portion engages its antigen, that sends an activation signal to the T cell, telling it to proliferate and kill that tumor cell. So a very simple and elegant idea that was actually first derived in the 1980s, but it took several decades to work its way through, all the feasibility issues of doing gene therapy in general. But as of about 6 or 7 years ago now, we started to see the first clinical trial results come through with this strategy, again in the CD19 leukemia lymphoma setting, where just really astounding clinical responses have been noted. So 40 to 90% complete response rate, meaning complete elimination of tumor, detectable tumor from patients in both children and adults with CD19 positive leukemias and lymphomas. This has led to rapid FDA and Health Canada approvals for this treatment, and it's rapidly becoming standard of care in this setting against these particular malignancies. As part of my day job, a bit of an aside, but also some context for our company, I actually oversee BC and in fact Canada's only CAR-T cell manufacturing facility. So this is the Conconi Family Immunotherapy Lab. It's based at our research center in Victoria at BC Cancer, part of PHSA. And we're using an automated system produced by a German company, Miltenyi. It's an automated CAR-T cell manufacturing unit that has it's a fully closed automated system that produces a CAR-T cell product on a patient-by-patient basis in 12 days and uses some very sophisticated bioengineering solutions to achieve that. Just some snapshots of our team. And so we have an open phase 1/2 clinical trial right now with patients being treated in Vancouver and Ottawa with our CAR-T product. So CAR-T cells are great. They've really, set it's a real watershed in medicine, the use of these genetically engineered cells. But as with any new thing, there's major challenges that remain. And so there are significant toxicities in many patients associated with this treatment. These CARs so far have shown a limited ability to penetrate into solid tumors. And so the response rate is much higher against liquid leukemias than against solid lymphomas, for example. And it's so far been difficult to safely target other cancers. There are also high manufacturing costs due to the need, at least with today's products, to produce them on a patient-by-patient basis and to give relatively large cell doses. So all of these problems could be mitigated partly or wholly by, again, this idea if we had precision tools that allowed us to control cell behavior. So what kind of cell behaviors would we like to control? Showing here just a generic T cell as an example. We'd like to be able to control their proliferation. We'd like to encourage them to infiltrate into tumors, to kill tumors. We'd like to be able to stop their activity if toxicities were arising or if the tumor was gone and it's time to go back to normal. We'd like them to form a memory compartment to protect against future relapse of the cancer. And in some situations, we'd like those cells, once they've done their work, to die, again, just so we can reset things back to normal. So decades of basic immunology research have taught us about the different cytokine signals that control these different behaviors. The issue, though, is if you are just to give a patient systemic infusions of cytokines, most of them come with very serious side effects. And so IL-2 is a classic example. It is used in a very limited way to trigger a T cell proliferation and killing in cancer patients, but it comes at a very high cost in terms of toxicity. So the idea that Marty and I rallied around together starting back in 2016 was to engineer selective cytokine receptor pairs that would allow us to deliver precise instructions just to the T cells of interest. And I'll explain a bit more about what that means and how we did it. So the idea here, and this is arose from a collaboration between my lab, Marty's lab, and Zymeworks, again, starting back in 2016 with funding from NSERC, was to take a natural cytokine receptor, shown here, and we'll just focus on the extracellular domain for the moment, and Zymeworks and Marty's lab working together introduced mutations into both the cytokine and the receptor, so-called co-evolved the receptors so that the cytokine would no longer bind to the native receptor and the mutated receptor would no longer bind to the native cytokine. But the two variant pairs, the cytokine and the receptor, would maintain high affinity for one another. And so this is a model of crystal structure of the cytokine and the receptor that we've used in our design. And this is just showing the interface between the cytokine and receptor. And essentially what was done, again, by introducing mutations into each, you change the knob and hole structure between the cytokine and the receptor to create a new interface with exclusive binding property. And so this was achieved. We've got the first, really positive results, I guess about a year and a half ago, and that led to filing our first provisional patent, which allows us now to send a signal selectively to cells that are engineered to express our receptor. The second innovation has been on the intracellular domain. We've created a toolkit of receptors with different intracellular signaling domains that allow us to send different messages to cells. Again, a proliferation message, killing, infiltration of tumors, slowing down, stopping, even dying. We've created this suite of intercellular domains to allow us to select what messages do we want to send to our T cells. And so that has led to a second provisional patent, which has been filed related to these different ICD solutions. So the way that this will work clinically, I'll illustrate in the next couple of slides. So whereas today's CAR-T cells have the CAR molecule that I talked about that allows recognition of the tumor and activation of the T cell, the idea here in the cancer setting would be to introduce or co-transduce cells so that they express not only the CAR, but also our inovokine receptor. And that would then put these cells under the control of our inovokine cytokine. So the way this would work clinically, a patient would come in, they've got their cancer, they're going to be treated with CAR-T cells. Now, again, the CAR-T cell product would be modified to have not only the CAR, but also our receptor. Those cells would be manufactured in a facility like the one we have in Victoria or any around the world, and infused into the patient systemically. Those T cells will circulate through the bloodstream, start to show up at the tumor site. And when we then add infuse our cytokine, shown here in teal, and that would just be through a subcutaneous injection, we can then send an activation signal to our engineered T cells, encouraging them to proliferate, to infiltrate into the tumor. We're working on designs that will have our T cells call out to the immune system and bring in other immune cells that can help with the anti-tumor response. So the goal would be to have a massive yet controlled immune response against the tumor. Tumor goes away, and at that point we can withdraw our cytokine, and the T cells will then be programmed to slow down and many of them to eventually die. And then the whole system can then reset itself back to normal in this idealized scenario. So this is my last slide, just to summarize what we have and what our future plans are. So again, a suite of receptors with engineered extracellular domains that allow us to achieve that selectivity, meaning to send signals only to T cells that are bearing our receptor. And then we also have this suite of intracellular domains that allow us to deliver specific instructions. I'm just showing one ECD here, but we actually have several different pairs that we can use. So there's even opportunities for combinatorial strategies where we'd be able to send different messages to cells depending on which of our cytokines we were to deliver to the patient. So in one example would be to put into the T cells a receptor that serves as a gas pedal, steps on the gas and promotes T cell proliferation, and another receptor controlled by a different cytokine that would be like a braking mechanism. So we could tap on the brakes when necessary to gain better control of those cells. In terms of applications, I've talked really exclusively so far about CAR-T cells, and we think that's a fantastic application, and that's the main one we are considering right now, especially given my interest and experience in the immuno-oncology space. But the same idea can be applied to engineered T cell receptors, which is a different targeting method, CAR NK cells, natural killer cells, CAR macrophages being developed by several groups. So a number of, we think, very exciting applications in the immuno-oncology space. We're also looking, though, at applications in inflammation, organ transplantation, and allergy, where CARs can also be put into regulatory T cells. These are inhibitory T cells that can be used then to dampen down overly strong immune responses in these other settings. So we see a great potential in this area as well. And then finally, just looking beyond the immune system at stem cell research, regenerative medicine in general, the idea, anytime you need to send a signal to a cell of interest, we think our technology can come in and help with that. And so we're looking at applications in these areas as well. So I'll stop there and point people to our website for more information, or feel free to send us an email if you'd like to follow up on any of these points. And thanks again for the invitation and for your attention and interest.
Thanks very much, Brad. Really appreciate you joining us today. We're running a little bit behind schedule, so I'll quickly go on to introduce Jonathan Polak. Jonathan is the CEO of Mesentech, and he works alongside Dr. Robert Young. He was the head of business development at Novateur Ventures, the founder and former CEO of DataPoint Diagnostics, as well as the co-founder of C7 Bio. Jonathan has a deep international experience in, in clinical development, financing, regulatory strategy, pharmaceutical partnerships, recruiting, angel investment, asset sales, and partnering. Having lived and worked in many countries, he is able to speak 5 languages. It's my pleasure to introduce and welcome today, Jonathan. Thanks for joining us.
Hi there. Thanks, Joe. That's quite an intro. Should have said something shorter. Anyway, thank you all. I'm going to share my screen here with you all. Okay, so what we are is a mechanism to deliver a drug into a specific tissue. And the tissue that we deliver drugs into is bone. And so the way it works is we have a proprietary bone targeting moiety. It's basically a vehicle that carries a drug to the bone. Well, it carries it in the body, and it only releases it when it's in the bone. And so what the rationale for this is, what's the purpose of it, is really to eliminate the systemic exposure of other organs to whatever this payload is. And so let me give you an example of one of the reasons why we needed to do this. And so in the history of bone drugs, we've had all of these ones that are above this line. They're all anti-resorptive, which means they stop bone from continually being lost. So these are all drugs for osteoporosis. The first really big success was Fosamax, which was developed at Merck. And then came a lot of other drugs of the same class until Prolia came along, which is an antibody. But they're all still in the same mechanism of action, which is to stop bone from continuing to be lost in an osteoporotic setting. But there were other attempts. And for example, 3 approved drugs that are actually have a label to reverse bone loss. And they don't really do a tremendously good job. This one does about 6 or 7%, and these are all comparable, but they all come with black box warnings. So the challenge has been, how do you get a drug into bone tissue while not exposing all the other organs to side effects that result in black boxes? So for example, Forteo and Tymlos have a black box warning around osteosarcomas, and Evenity has black box warning around strokes. Similarly, some of the failed programs, this one at Merck, also developed by Bob, the founder of Mesentech, Odanacatab, was found to have a slight increase in strokes after a 16,000 patient phase 3 trial that costed well over $1 billion. And thus, Merck never filed an NDA for that drug because it would have had in black box as well. These 2 in particular are highlighted because both Pfizer and Merck had programs targeting the same pathway, which is actually the pathway that we're targeting too. So these 2 programs were actually, this one was being led by Bob when he was still at Merck, and this one at Pfizer was being led by David Thompson, who's the chairman of our scientific advisory board. Now, these are really interesting drugs. They're actually mimetics of prostaglandin, which of course is a natural occurring signaling hormone in the body. And they were the most anabolic compounds, meaning the ones that reverse bone loss and build bone more than anything else that has ever been tried. But they had a really difficult profile in that they caused very severe and precipitous hypotension, meaning participants, subjects would lose consciousness during the drug administration. That's how severe that hypotension was. So what Bob decided to do is he took this drug here from Merck that we know obviously very well and conjugated it to create a drug conjugate where the Merck portion, which we know works very well, but has all these side effects, is now attached to a carrier vehicle, which is this bone targeting moiety that I talked about at the beginning. So the basic premise is this whole thing is connected. You administer it to the patient. It connects, it binds to bone. It's actually a phosphate, and phosphate, this piece has 2 phosphates on it, and it has a high affinity for bone. And then with a certain half-life, this connection here, this bond breaks. This has certain anabolic effects, and the mechanism of action is mesenchymal stem cells, as well as so stimulating them to differentiate into osteoblasts, as well as protecting osteoblast progenitors from apoptosis. Basically, you build bone like crazy. I'm going to skip this in the interest of time so that we can end a little bit quickly. But essentially, one of the challenges in making bone is to disconnect the balance, these 2 processes. So building bone and resorbing bone are normally connected, and all of the drugs kind of have a hard time of increasing one without the other. What this drug, this prostaglandin mimetic, has been able to do is actually separate them. So what we have done, actually really important, let me see if I can turn this on. Can this work? No? Okay. What, oh, there it is. So we did the experiment in dogs, and we demonstrated that we have absolutely 0 hypotensive effect. And this is a relatively new finding as of April of this year. So that is a really important finding for us, because we've essentially proven Bob's hypothesis. They can take a compound that has a really well-known effect, but mitigate its side effects while preserving its pharmacological efficacy. We have a deal with a major pharmaceutical company who really doesn't want non-COVID news going out right now, so they wanted to keep themselves anonymous for now until COVID is over, as well as an investment by CQDM and a different pharmaceutical partner that they're working with. We also have recently finished the first part of a trial in osteogenesis imperfecta. That's more commonly known as brittle bone disease. It's a disease that you often diagnosed in childhood, where kids have a lot of fractures. So we'll present some of that data at the American Society of Bone Mineral Research in September, and we're proceeding with an orphan drug designation for that. Manufacturing, which for a conjugate drug is actually not trivial, is proceeding very, very well. And we have all of our GMP inputs complete, as well as all of the GMP material will be ready by September, October timeframe. So we're on track to have an IND filing by 2021. And this is kind of like going forward that we'll be pursuing also bone side effects of muscular dystrophy, in particular Duchenne. And I just wanted to leave everyone off with, my gratefulness to Bob Young, who's not on our call today, but Bob is the only reason that Mesentech exists, and the reason I came to work here. He's really an exceptional drug development, die-hard drug hunter who is, someone that I have the privilege to work for, and I should pay him for the privilege of working for him. He's really an exceptional drug developer that is still making drugs into after a 30-year career. He retired to start another career as an academic, and he just can't stop making drugs. So I'm very grateful to Bob, and I'll be around for Q&A in one of the breakout rooms if anyone's interested. Thank you.
Thanks very much. That's really interesting, and congratulations on all the success that you have been having. Some that you can talk about and some that is confidential. So there is a poll on the screen right now that if you could take a couple of seconds to just answer it, it would be great. We're trying to find out about future topics. I want to thank Joe and Blake's again for continuing to host and ride this journey with us as we adapt to holding virtual events. We are going to move to the breakout rooms right now. So you would have received an email if you registered for the breakout rooms sometime over the last half hour that came from LSBC, and that is the link to the breakout rooms. So once these polls are finished, what we will do is close this off, and we will see you in the breakout rooms. And I believe Dr. Nelson and Jonathan are going to be joining us as well. So you can ask questions to them as we do that. Okay, thanks. With that, I'm going to sign off and move into the breakout room.